Hello, I'm Kelly. I'm one of Mel's PhD students, and she's asked me to talk to you today about antimicrobial resistance and biocides. So just a quick introduction to myself. I'm Kelly. I'm in my second year of my PhD. Before that, I did my bachelor's in applied biology and then my master's in molecular and cell biology, and now I'm working on my PhD. And it's entitled Development of Antimicrobial Impregnated Catheter Coatings to Prevent Uropathogenic Escherichia coli Infections. But I'll tell you a little bit more about that in a bit. So this is what I'm going to talk to you about today. I'm going to tell you a bit about antibiotic resistance, what it is and why it's a problem. And then I'll tell you about what I'm doing, my research into uropathogenic E. coli and the biocides and quorum sensing inhibitors I'm using rather than antibiotics, and give you an update on my results so far. So, antibiotic resistance. What is it? Antibiotics are chemicals which stop bacterial infections. They do this in really specific ways, attacking a certain part of the bacteria. So they do this to kill the bacteria, which stops them growing and stops the infection continuing. So antibiotics, they normally come from nature, but we do have synthetic ones as well. And there's lots of them, and each of them are unique, and they attack very specific aspects of the bacteria. But then we've got resistance. Bacteria develop these ways of surviving the antibiotics attack on them, and it does this in several ways. So one, it can just kick the antibiotic back out of the cell. Next, it can change the thing that the antibiotic acts upon. So if it acts upon an enzyme, it'll change the enzyme slightly so the antibiotic doesn't work anymore. Or it will avoid that pathway altogether. So if the antibiotic attacks this, bacteria will find a way to avoid it. The problem with bacteria is that they grow rapidly. They can mutate easily as well. And once a mutation arises, it can very quickly spread through the whole of the strain that's available there. So, with an antibiotic infection, you have the bacteria, so you treat it with an appropriate antibiotic, which will kill the bacteria. But if this bacteria mutates in some way, it can stop the antibiotic from working. So this new strain of bacteria can proliferate. So we find another antibiotic to treat this one, and it kills the bacteria for now. But then, what if another mutation occurs? So this one has lost all of its pili. The antibiotic stops working again, and we need yet another antibiotic to kill it. So you can see how it can cycle, and bacteria can collect all these mutations. So more and more antibiotics are needed to kill what could once be treated with just a single antibiotic. So what is antibiotic resistance and why is it a problem? The problem is that widespread and inappropriate use of antibiotics are exposing lots of bacteria to antibiotics. They're putting these selection pressures on them. This is causing them to mutate, and then this antibiotic no longer works against that community of bacteria. Another problem is the fact that we have a limited number of antibiotics, and in some cases we're running out of them and we only have last hope antibiotics left to treat some infections. Antibiotic resistance spreads really quickly as well. Bacteria can spread to one another, they replicate rapidly, so one bacteria with a mutation can replicate lots, and then you've got exponentially more bacteria. Or it can also pass these genes for the resistance to other bacteria, not only surrounding it, but other species of bacteria as well. And they can collect all these mutations, so they can be resistant to different types of antibiotics all at the same time. So this is really concerning, because the more bacteria that become more resistant to more antibiotics, the harder bacterial infections are to treat. Right now, this is from 2014, at least 50,000 people in Europe and the US die each year from antimicrobial resistant infections. and it is much higher than that in the rest of the world. Right now, it's estimated that around 700,000 people die from antimicrobial resistant infections. But the scary thing is it's estimated to be up to 10 million people by 2050. My research is to create a coating to go on a urinary catheter and prevent E. coli infections. So this is a urinary catheter. It goes up the urethra, into the bladder, and held in place with this balloon. This is a Foley catheter. The problem with this is, as useful as it is to treat urinary incontinence and urinary retention, the problem is it can become colonized by bacteria quite easily. They can grow on the outside surface of it or here in the lumen. If they're infected from the outside, they can climb up into the bladder and continue climbing to the kidneys, causing kidney issues, or get into the blood. They can also be introduced as it's inserted, so it can carry it straight up into the bladder. And the consequences of this are really high, ranging from fevers and pain, all the way to blood infections and death. So you can see why it's important that we try to minimise the number of infections which are caused by urinary catheters like this, because they are really commonly used. So I'm working against uropathogenic E. coli. These cause about 40% of the catheter-associated UTI infections, so quite a lot of them. The thing with them is that they're gram-negative, they always are. They've got an inner membrane, a peptidoglycan layer in the middle cell wall, and an outer membrane as well. They have some of these features that Mel's been telling you about, the flagella, fimbriae, pili. They're able to form biofilms. They communicate to one another through quorum sensing, and some of them can secrete toxins like hemolysin as well. Another feature of the bacteria I work with are that they're extended spectrum beta-lactamases. So this means they're resistant to penicillin, cephalosporins, carbapenems, and many more antibiotics. And the thing that makes them resistant is that they've got the ability to break down this lactam ring structure here, this square ring. So what do beta-lactams generally do? They inhibit the cell wall synthesis. And it does this by blocking the cross-linking between the peptidoglycan units. So normally the two units are linked together by this cross-link between amino acids. But in the case of using the beta-lactams, this inhibits the enzyme which does this, so that the cell wall cannot form properly. The beta-lactamases, they've got this enzyme, the beta-lactamase, which breaks this lactam ring, and so the cell walls can then form normally and the cell can continue to grow. So for part of my PhD, rather than using antibiotics, so you can see that especially the beta-lactam based ones aren't going to work against these bacteria. So I'm using a few other different types of molecules. One of them are the biocides. So these, unlike antibiotics that target only one aspect of the bacteria and inhibit cell wall synthesis, instead they attack several parts of the bacteria all at once. So they can break down, say, the lipid membrane, but also attack the DNA and inhibit enzyme function as well. That's what one of mine does. I'm also working with quorum sensing inhibitors. So quorum sensing is a way of bacteria to communicate to one another. They let each other know that they're available in the location, so that they can then, once there's enough of them, change their gene expression so that they can be more pathogenic. What quorum sensing inhibitors do is they block this communication, so the bacteria still think they're on their own, and it's not worth becoming virulent, as this will alert the host, the human, to their presence and attack them with its own immune system. So what I'm doing is I'm combining the biocides and quorum sensing inhibitors and seeing what that does. So my My hypothesis is that combining these biocides, which attack several aspects of the bacteria, with the quorum-sensing inhibitors, which stop the community-focused virulence side of the bacteria, that we should need lower concentrations to kill the bacteria, and therefore we'll be able to more safely use it on a catheter coating, so it won't cause too many problems for the patient, and we stop these bacteria forming biofilms or even just surviving long enough to cause an infection. I do this it's a standard way in microbiology. So we've got the bacteria growing on a plate, we grow them up overnight, and then we add them to the wells of this plate. In each individual well, there's a different concentration of either biocide or quorum-sensing inhibitor, and in some wells there's a mixture of both. And we've got those at a whole different range of concentrations, so that I can measure how much it changes by. So we see if they grow in here, that means the wells will become cloudy if the bacteria grow. And then we also plate them onto agar to see if they then grow up as well. And we can see if it kills them or not, or just inhibits their growth. And I also do this with biofilms as well. So what have I found so far? Well, in terms of inhibiting bacteria, so stopping them from growing, we do need a lower concentration of each. So the amount of PHMB, this is polyhexamethylene biguanide. It's a biocide which attacks the membrane of a bacteria, and it also attacks the DNA as well within the bacteria. I saw that it needed 2 micrograms per mil to kill the bacteria, so to inhibit the growth of the bacteria. And with the cinnamaldehyde, this is the quorum-sensing inhibitor, so this stops them talking to one another. It needed 500 micrograms per mil to inhibit their growth. But when we join them together, so this is the amount of PHMB when it's with cinnamaldehyde and the amount of cinnamaldehyde with PHMB in it, we see a big reduction in the amount needed. We see the same thing with killing the bacteria. So as you can see, the amount needed to kill the bacteria is higher than just stopping the growth of the bacteria. So we've got 7.5 micrograms per mil. But when in combination with cinnamaldehyde, same thing with the cinnamaldehyde. Again, it was almost 2,000 micrograms per mil to kill the bacteria this time, so quite a big jump up, but less than 500 to kill them when combined with PHMB. So with the biofilms, we thought this would be more interesting because forming biofilms is generally related to being able for the bacteria to talk to one another. The quorum-sensing inhibitors should affect this, and we see that it does. So another aspect of biofilms is the fact that they can need hundreds to 1,000 times the concentration to kill the bacteria, to kill the biofilm than it does to kill the bacteria. And as you can see here, we're at 170 micrograms per mil to get rid of a biofilm, whereas previously it was 7 micrograms per mil. So it's quite a significant increase there. And same with cinnamaldehyde, again a higher concentration needed, not too much though. But again, when we put these in combinations, we drop them way back down. The big benefit of this is that all these chemicals, no matter what, are toxic to humans as well. So if we can lower the concentrations of these to less than 40 and about 500 micrograms per mil. So there's a lot of benefits to this. So we saw that combining the biocides and quorum-sensing inhibitors together reduced the concentrations needed to kill them, and they killed them more effectively. So that's really advantageous. And I saw this not only with the PHMB and cinnamaldehyde like you've seen there, I'm also testing several other biocides, and I'm seeing generally positive results like that. So my next step is to combine the biocides and quorum-sensing inhibitors together in a coating on the urinary catheter. So I'm working with a coating which is called Sol Gel, and this is a liquid which I can add the biocides and quantum-sensing inhibitors to, and it'll dry on the outside or inside of a catheter. What I've got to do then is see how much of the biocide and quantum-sensing inhibitors leach from the surface into the surrounding areas, how much stays on the catheter, and see how this affects the bacteria when exposed to it. Does it stop the bacteria attaching to the surface of the catheter? Do the antimicrobials which have leached off just kill the bacteria which are in the environment? And can we stop the biofilm formation, which is the biggest challenge? So, in summary, antimicrobial resistance is a major problem. We could see up to 10 million deaths per year associated with this. And this is a problem because bacteria can develop resistance to antibiotics so easily, and they can pass this amongst them. Some of the alternatives which are being looked at are this use of biocides and quantum-sensing inhibitors like what I'm doing. It means that the bacteria not only need one mutation to develop resistance, they need several at the same time for them to be able to overcome it and survive in the presence of the biocides and quantum-sensing inhibitors. My research so far suggests that combining these together is effective, and so I'll continue to work on that. Thank you.